All right. Hi, my name is Milan Radovic,
And I'm Brian Schneider.
and we co-lead the IU Health Precision Genomics program, and today we're giving you a video recorded lecture on cancer genomics and precision medicine. So let me share my screen. So I'll let Dr. Schneider start off, talk a little bit about precision medicine.
Yeah, so the idea and concept of precision medicine is really not a new concept. I mean, I think in the world of medicine, we've been thinking about trying to tailor our therapies around an individual for many, many years. But I think in the last couple of years, with the advent of some of our newer technologies like genomic technology, proteomic technology, and others, the real concept of really tailoring our therapy to cancer patients has really hit mainstream, and something that we've been passionate about here. So in terms of thinking about precision medicine, what we've been passionate about is really thinking about this both for providing good clinical care, but also at the research end. And here at Indiana University, there has been a big push toward translational work that really focuses on bringing precision medicine to our patients at multiple levels.
So I don't think any talk on precision medicine can really start without taking a trip down memory lane and really start from the discussion of the Human Genome Project. And when it comes to precision medicine, DNA-based precision medicine by far has led the way. Why? Because the unique makeup of our individual genomes really can implicate what diseases we experience, what toxicities we experience, how we respond to different drugs that we're administered. So as many of you know, the Human Genome Project was a true feat, a project that took about 11 years and $3 billion to sequence the first 3.2 billion letter code that makes up you and I, first released in June of 2000 at the White House. What's happened since the Human Genome Project has been advances in sequencing technologies. And just like cell phones, sequencing technology has gotten cheaper, faster, and smaller, such that back in the days when we used to sequence DNA using gel electrophoresis, we could do it on the order of thousands of bases per day. But now moving into the late 2000s with the advent of next generation sequencing, be able to sequence on the order of billions of bases per day or multiple genomes per day. The newest machines currently on the market can sequence anywhere from 48 to 96 genomes within 3 days. So this technology has gotten cheaper and faster and smaller, and now it can be enabled for everyday clinical care. The future is continued advances in technologies, and technologies such as nanopore sequencing will continue to miniaturize the technology, but also to speed it up such that we can envision a day when genomics and precision medicine is actually done at the bedside, where maybe not even needing specialized laboratories to do sequencing, but one that could be done as just as simple as a CBC. So when it comes to precision medicine, there's no field that probably has more advances in precision medicine than cancer. And really for one main reason, is because cancer in itself is a disease of DNA. Cancer is caused when certain mutations occur in the genomes of normal cells that transform them from going from normal cells to becoming malignant. This process occurs through our lifetime, such that mutations are incorporated, can be incorporated randomly. So these are mutations that occur in the genomes of normal cells when cells divide. Our machinery that replicates DNA is really good at what it does, but it's not perfect. So mutations are incorporated in the genome every time we have a cell division. But we also know that certain environmental and lifestyle exposures can also incorporate mutations to the genome. So things like smoking and sunlight can mutate DNA. And we also know that patients can be born with mutations that predispose them to developing more mutations. And so things like BRCA1 and BRCA2 that predispose these genomes to become more mutable. What we've also learned through advances in technologies and research is that cancers can be highly heterogeneous.
Yeah, so this is an article that was published now almost a decade ago, which I think really for us cancer researchers and clinicians became one that was pretty horrifying. And that if you look at a variety of areas within a tumor, and also if you look at different metastatic lesions, sometimes the genomic makeup or the mutations that we see are different. And for those of us who hope for the silver bullet of finding a target that we can hit with a drug, this becomes a very, very complicated system now.
Well, we also know through lots of broad-based genomic sequencing is actually the number of mutations a particular cancer genome can have can vary widely. And so this is, again, another landmark paper published in Nature in 2013 that really asked the question, well, how mutated are cancers in general? And when you look on the right side, these are the cancers that tend to be mutated by exposures. So things like melanoma due to sunlight, and lung cancer and bladder cancer due to smoking. And then on the left side are your cancers that are the least mutated. And these tend to be your pediatric cancers like rhabdoid tumors and Ewing sarcomas. And then the majority of cancers being somewhere in the middle. But again, the number of mutations and how mutable a cancer is varies dramatically by orders of magnitude. So when it comes to studying cancer genomics, this is the very oversimplified overview, but we sequence a tumor genome and identify its variants. We sequence the normal matched germline, identify the variants, subtract the germline from the tumor, and we have the catalog of somatic mutations. And somatic mutations in general is what we're looking at when we're thinking about largely for intent to treat. But the germline mutations, as you know, are also quite important, which we'll highlight later in the talk. So again, if we continue to go down memory lane, these advances in sequencing technologies and bioinformatics really came to a culmination with this paper published in November 2008, which was the first cancer genome ever sequenced published in Nature of a cytogenetically normal acute myeloid leukemia. This was about the 7th whole genome at the time ever published, and again, the first cancer genome. What actually wasn't realized when it was first published, but then in subsequent editorials, was this actually was the first female genome ever sequenced and published and released. During that time, this was really the heyday during the advent of cancer genomics, such that sequencing centers that had enough sequencing capacity could sequence one cancer genome and actually publish a major paper in journals like Nature and the New England Journal of Medicine. And probably one of the most famous papers at the time was actually the sequencing of a small cell lung cancer genome. Again, as many of you know, these cancers are almost exclusively caused by smoking. And this paper was famous for 2 reasons. The first reason was it was the first paper to ever utilize what we call a Circos plot. So the concept of how can you depict an entire genome in a single figure without publishing 3 billion letters in the supplementary data. And so what you have here in this Circos plot is it gives you a snapshot of how perturbed this particular cancer genome is. So on the outer circle, you have your karyogram. And then as you move inside with every concentric circle, it tells you a little more information about the particular genome. So we learn about its copy number variation, the number of point mutations, the number of translocations here. So the green are your inter-chromosomal fusions, your purple are the green is your intrachromosomal fusions, your purple are your interchromosomal fusions. And just as an overall picture, what you can see is that this particular genome is pretty perturbed, and we would expect that from a genome caused by smoking. But probably what made this article make it into the New York Times and the Wall Street Journal at the time was they had the smoking history on this particular patient, and they were actually able to calculate that one mutation was incorporated into the genome for every 15 cigarettes smoked. Now, we've known since probably at least the 60s or 70s that smoking was mutable and cancer-causing, but this was the first time that we could actually put a number to the mutability of smoking in cancer genomes. So after some of these several seminal papers, the National Cancer Institute got involved and really said we need to do this on a much more broader scale and launched a really ambitious initiative called the Cancer Genome Atlas. The Cancer Genome Atlas, its original goal was to sequence more than 10,000 tumors and 24 tumor types by 2015. It ended up extending the project to go over to several thousand more tumors that went into about roughly 35, 36 tumor types. But really an opportunity to do comprehensive omics sequencing using a variety of technologies listed here, with the express goal to create a comprehensive public catalog of all significantly recurrent genomic alterations. For those of you who are doing your thesis research or your research project in cancer genomics, I'd like to point out there's great resources out there for your research to access this. It is freely available. The 2 best resources are the NCI GDC data portal or Genomic Data Commons. This is where you can download the raw data from TCGA as well as a lot of other cancer genome consortiums. But for those who may not be as bioinformatically savvy, we have this very user-friendly tool, cBioportal, that's run by Memorial Sloan Kettering, that really allows you to quickly analyze data from publicly available consortiums to ask the questions such as, how does a particular mutation associate with survival? Or what's the prevalence of a particular mutation in a particular cancer type? So again, for those of you doing your research, I highly would encourage you to look at this particular website. So I'll actually hand off to Dr. Schneider to talk a little bit about what are some really cool advances now on the horizon with sequencing technology.
Yeah, you know, as we think about the need to do or the desire to do sequencing to help identify drug targets in particular, one of the real downsides is that to do it well, we have to biopsy a tumor to really identify, do the sequencing, and find the target. This can lead to some pragmatic issues in that sometimes the biopsies are not simple, and sometimes they put the patient at great risk from a medical standpoint. And so one of the golden grail ideas would be the ability to really do a non-invasive approach to do sequencing of the tumor. And so this brought forth the idea of doing blood-based biomarkers with things like circulating tumor DNA. And what we know is that as a cancer lives and dies on its normal life cycle, as it apoptoses, it sheds its DNA into the bloodstream. And so the ability then to take a blood sample separates one's normal DNA from the tumor's shed DNA, and then to subsequently sequence it has been a great advance in terms of identifying very small concentrations of tumor and subsequently finding targets.
So ctDNA analysis has really made its first headway in the concept, as Dr. Schneider mentioned, to be able to look at tumor DNA mutations without having to do a tissue biopsy. But where it's really probably moving forward in a very hot area is this concept of being able to use it to detect what we call minimal residual disease. So this ability to actually see if cancer is present in a patient that we hope would be cured from their initial therapy. So if we take an example here of a patient with breast cancer who only has disease in the breast, and if we were to surgically remove that breast tumor, one could simply say, well, this patient's cancer-free. We've removed the tumor from the breast. We don't see any disease on any CT scan, MRI, or physical examination. However, what we do know is that there's a proportion of these patients where that cancer is going to come back. We call this relapse. So one could potentially, after surgery, take a blood sample, and if a patient is negative for ctDNA, those patients may not need any further therapy. Maybe there is no micrometastatic disease present. However, if we take a blood sample and we detect this tumor DNA in the circulation, even though we can't see it on standard physical examinations, now we worry that this cancer is still present, that it has metastasized, and these patients will relapse. These patients are really high risk of their cancer coming back. So here's a very interesting example of a case of a patient where this analysis was performed. So this was a patient with triple negative breast cancer whose tumor harbored a p53 mutation. And what you have on the y-axis is the allele frequency of the particular mutation in the plasma. And on the x-axis is the time point. And so what we did was, before surgery, we could adequately detect the presence of the mutation. And then just after surgery, at the first time point, actually we do see the mutation. And then the detection of that mutation goes away while the patient was on therapy, in this case with a chemotherapy called cisplatin. However, after the patient came off the cisplatin, the allele frequency of that particular mutation began to rise again, and unfortunately the patient had a clinical recurrence and unfortunately succumbed to their disease. But future trials that can really harness the power of ctDNA to really risk stratify those patients who are at high risk of relapse really represent the future of how we do trials in this curative setting. So we're going to transition now. We talked a lot about Cancer Genomics 101. We talked a bit about research. Now what we really want to focus is how we use this for clinical care.
Yeah, so, you know, again, probably about 5 or 6 years ago, as we began to see some of the advent of genomic technology in terms of being able to catalog many of the major tumors and finding what we considered good drug targets, Dr. Radovich and I really wanted to set out to start to take this sort of technology and move it into actually making care better for our patients with cancer. And of course, one of the early concerns of sequencing was the cost. Obviously, this was a very expensive technology in the early days, but as Dr. Radovich mentioned earlier, like cell phone technology and computing technology, because of the advances in technology, the price also began to become really more affordable. The other is speed. And as he mentioned, it took over a decade to sequence the first human's genome. Again, very similar with computing technology. The ability to do this in real time for clinical care became markedly better as well. And so, the idea here is to do this in a way that we can make an impact on patients' care. You need to be able to turn these results over in a few weeks, which is now a reality. And then finally, of course, other issues of implementation, meaning putting this in a house that is regulated by CLIA so that these results can be used to actually make a clinical decision. And then finally, some other obstacles to overcome is how do you get these sort of things reimbursed? For many of our drugs, we're identifying drugs that wouldn't necessarily be on label for that specific tumor, but rather be appropriate for that specific mutation. So how do we identify ways to actually get the drugs covered so patients aren't left with massive health bills? And then finally, trying to put this in a way that many physicians who are really spend the majority of their career understanding drugs that work for a specific disease now begin to help them understand complicated genomic technology. And again, one of the ways that we were trained as oncologists over the years was thinking about treating a disease based on where it comes from. So we are often trained for tumors that arise in the breast. We treat that as a breast cancer, so with a variety of different drugs, which are often very different from a tumor that happens to arise, say, in the ovary or in the colon. What we've recognized now is that many of the fundamental changes that happen in the genome of these tumors are very different within the tumor. And interestingly, some of the changes we may see, for instance, in a breast cancer may be shared by mutations seen in ovarian cancer. So really what this does is allow us to pop open the hood of the car and see what's happened to the engine rather than thinking about, treating this again as a disease that starts in a specific place. So it's allowed us to really personalize our treatments. So this is a slide that we've both used for probably well over a decade now, thinking about what makes good precision medicine. And here as we think about precision medicine, it's really trying to do two things. One is to identify a group of patients with a disease and treating them with a drug that will optimize benefit, minimize toxicity, but be able to do that before we ever treat the patient. So it's really, again, identifying markers that guide our therapy before we ever expose the patient. And I think the second fundamental principle is, again, not only the optimization of benefit, but also thinking about minimization of side effects from those given drugs. Go ahead and
yeah,
with this
so
one.
This is a horribly small list of all the potential drugs that could be, they're guided by genomic markers. Interestingly, now almost every month we have yet another FDA-approved indication. But just to give you a flavor of some of the ones out there, things like the BRAF V600E mutation, the drug vemurafenib, which was a small molecule designed to inhibit that particular mutation, first in melanoma, now also in thyroid cancer. Probably the most classical example is gene amplification of HER2 in 15 to 20% of breast cancers using an antibody that targets gene amplified HER2 receptor. We also know that SNPs that we're born with that influence how we metabolize certain drugs can influence the side effects we experience. So there's a rare mutation in a gene called UGT1A1 that's involved in metabolism of irinotecan that patients who have a particular SNP develop a pretty debilitating side effect. And it goes on. And so with markers and then with platforms, what we're seeing is a continued development of the concept of how we use genomics to really guide and tailor therapy for individual patients. So what we really wanted to get when we do this genomics and informatics is to get responses like this. I don't know, Dr. Shah, you want to open this page?
Yeah, so I mean, I think every institution has their kind of favorite case of where targeted therapy did something that you wouldn't expect. But this is one of our favorite. This was a gentleman in his 50s who had a really bad form of cancer known as metastatic anaplastic thyroid cancer. These are tumors that begin in the thyroid, and whereas most thyroid cancers are reasonably easily treated with surgery, this is one with a mortality rate of essentially 100%, with an overall survival that often is somewhere around 6 months or so. These tumors are highly refractory to standard therapies, which are often platinating agents and anthracyclines. So we saw this patient and did sequencing, and he was found to have a BRAF V600E mutation. So I think, as mentioned before, this is a really classic mutation seen in melanomas. The drug that blocks this pathway is vemurafenib. The patient also had PD-L1 positivity. This is a marker to suggest stimulating the immune system may be a reasonable strategy. So this patient was treated initially with the vemurafenib to block the BRAF mutation and had a beautiful response to therapy. Interestingly, though, after a short period of time, began to develop a recurrence in some of his tumors. And so his treating oncologist then added an immune therapy using a checkpoint inhibitor to really unleash the patient's T cells against the tumor. And the patient had a marked response. In fact, all disease went away and did well now for over a couple of years. Yep. So this is an example of a dramatic response of a patient doing well, which would not have done had he not been sequenced.
So what you've seen in more recent history is really the development of programs at academic medical centers that are dedicated to providing genomics for patient care. And Dr. Schneider and I are biased, but we believe our program really sets the standard of how it should be done, and we'll be delighted to teach from that perspective. You want me to? Sure. So the IU Health Precision Genomics program was started in 2014 by Dr. Schneider and I, with really the concept of a clinical program with an academic base, meaning we wanted to take all of the know-how and knowledge that has been developed at the IU School of Medicine in genomics and informatics and oncology and precision medicine, now dedicated to patient care. So our patient population is primarily tumors that are refractory to standard options or rare tumors where there are no standard options. Again, we use cutting-edge next-generation sequencing, in particular whole exome and whole transcriptome sequencing. We also look at host genomics to look at both germline toxicity SNPs as well as germline mutations for cancer predisposition. We make our therapy selections based on the strength of association of a particular biomarker with clinical evidence of efficacy, and always consider the patient's prior therapies and comorbidities and overlapping toxicities. As you can imagine, we are also very passionate about driving clinical research. As I'll talk about maybe just a little bit later, is that pharma is very interested in finding patients who have particular alterations in which new drugs are being designed against those key vulnerabilities. And we want to be able to provide our patients the best drugs available on the planet that can go after their particular genomic vulnerability. And then we obviously want to learn from the data we generate. So we're going to biobank specimens and the genomic data for translational research. So this is the patient journey for a patient coming through our program. Our referrals are going to come from medical oncology, and patients primarily see us for 2 visits, where on the first visit, they're going to get an intro to Genomics 101. They're going to get a full germline pedigree, a 3-generation pedigree performed, and they're also going to have an H&P to deem them appropriate for sequencing. If tumor tissue is available, we'll obtain that. But in many cases, Dr. Schneider here is going to order a new biopsy of a tumor lesion. The sample gets sent out for somatic and germline sequencing, and those results come back where they're reviewed by our team and then goes to a molecular tumor board. It's probably one of the most favorite parts of this program is our molecular tumor board, comprised of over 20 IU faculty and staff from a variety of disciplines, where we spend time going over each patient, looking at their prior history, the prior therapies, the genomic results, and really trying to come to a decision of what we should do next. This can include affirming an FDA-approved on-label drug, considering a off-label or what we call non-standard FDA-approved drug, or recommending the patient to an early phase clinical trial. As I mentioned, the tumor board, again, is probably, again, our favorite piece of our program. It is a great group of people that get together every Thursday morning at 8 a.m. to really spend time and each one providing their own unique input into the case for each patient. I should state that after our tumor board meets, we bring the patient back for a second visit where we go over the results with the patient. So we do a layman's education of their particular genomic alterations. We go over the drugs that we're recommending, how they work, if there are any potential side effects. If the patient carries a germline finding, our genetic counselor will go over the germline findings with the patient, encourage cascade testing and medical management. And then we also, if there's a clinical trial, we'll give all information to be able to match that patient to a trial, assess their eligibility, and then hand them off to the appropriate coordinators.
Do we hit incidentals over?
Yes. So as you can imagine, and I won't go into much detail with this, when you're generating this huge amount of data, it takes some serious computational resources to really take a genome and try to figure out what is in that genome we want to target, what is therapeutic. And back in the days when next generation sequencing precision medicine started, the tools were absolutely terrible. And so this is what it looked like. We called it the black screen of death. You would just have these tools that were really antiquated, they were slow, they never really provided useful output. But as we advanced in the research informatics technologies, things got quicker, faster, much more types of outputs being produced. Again, I'm just going to be very cursory in this, but when you can think about that when you have a tumor genome and its transcriptome, just the sheer number of endpoints that one can look at when it comes to trying to determine what is a potential therapy is almost virtually endless. For example, we look at markers for immuno-oncology. So what particular immune therapies may work for a patient by looking at their tumor mutation burden, their microsatellite instability, by the gene expression of a variety of immune checkpoints. We look at DNA repair because we know that tumors that are impaired in their ability to repair DNA may be sensitive to classes of drugs that target that. So we look at their somatic and germline DNA repair genes. We look at mutational signatures, so we can actually tell from the tumor genome sequencing how that tumor was mutated, how it was scarred. And in this case, we can tell without even knowing anything about the patient, whether their tumor was scarred by a BRCA1 or BRCA2 mutation, or by smoking, or by sunlight. We can look at their cytogenetics and look at their genome-wide LOH and their ploidy to be able to find particular vulnerabilities. Again, and it continues to go on. Mutations, copy number, gene fusions, pharmacogenomics, HLA type, neoantigen expression, so on. So when it comes to really thinking about what are the endpoints we kind of look, really our only limitation is the statistical package or the informatics know-how to how to do it. We, again, will always go to every advance possible to be able to find a particular therapy. Probably one of my more favorite tools is site of origin prediction. So this is actually taking the transcriptome data that we do in our patients and actually determining the site of origin based by comparing it to a large database of other cancers that have been sequenced. This is really helpful when we get a case that comes to our clinic that has a diagnosis of cancer of unknown primary, and we don't know exactly, the oncologist doesn't know may not know what the right treatment to use. And so very easily we can take the transcriptome data and actually match it and figure out its particular origin, again, without ever looking at the histology or the background of the patient. So I think, a big question is that when we do this genome sequencing of a particular patient, how do we define what's clinically actionable? So just because there's a bunch of mutations there doesn't mean that each one has a drug. And so in our program, we set this, here's our criteria. Our first criteria is that a biomarker has to have clinical evidence of association with response or resistance to a particular therapy. And we stress clinical evidence, meaning there's got to be something in the literature that says this has been clinically associated with response or resistance. Now that clinical evidence is going to range widely. It could be from level 1, which is a prospective randomized clinical trial, down to what we call level 2-3, which means retrospective analyses of completed trials. And truth be told, the majority of the evidence out there is retrospective, but definitely helpful. While we may use data produced in cell lines, mice, dogs, and primates, and so on, they do not count exclusively. They may be used to support a particular association, but never used exclusively. The second point for what is clinically actionable is that a genomic aberration enables enrollment onto a biomarker-driven clinical trial. As I mentioned before, we really want to get patients access to the most cutting-edge medication available on the planet that may match their particular alteration. So we really encourage patients to enroll on clinical trials when one is available. Now, with a clinical trial, here, you don't necessarily need clinical evidence because the trial is generating that clinical evidence. But usually these trials are backed by a large body of preclinical data. Again, and very importantly, we will never support the off-label use of agent based on preclinical data alone. You always have to have a preponderance of clinical data to support the use of an off-label agent. So when it comes to databases available to figure out what's actionable, there's a few really cool ones out there. Probably one of the first, actually the first that ever came out was My Cancer Genome, done by Vanderbilt. Very user-friendly, intuitive interface to look at whether a particular mutation is associated with the drug. MD Anderson has their personalized cancer therapy database. WashU took an interesting approach called Civic. It's evidence curation by crowdsourcing. So Civic is a database where anybody can go to it and deposit their own evidence, and then it's curated on the back end by a set of curators. Again, kind of taking the concept of how we can use social media and crowdsourcing to be able to advance our knowledge. But one thing we've done here at IU that I'm quite proud of is really try to develop a cutting-edge informatics platform that takes this evidence that's available out there in the public domain, but also incorporates our own evidence that we've generated over many years. So within our own program, we track about 21 different journals and meeting abstracts to curate our own evidence database as well of a gene-drug association. But we also want to have a database that can take all the data we've generated on our patients, as well as data from the EMR, and be able to put it into a single repository that we call the Precision Health Cloud. And what's great with this particular repository is that we can quickly identify patients for clinical trials. We can analyze their genomes for actionable alterations. We can actually soon engage patients with surveys of PROs. And as you know, overall, as we see the precision medicine move forward, what is going to be absolutely needed is informatics platforms that allow us to analyze this, because human beings, just the sheer amount of data is not capable by individual human beings. We have to have tools that are able to automate and scale these sorts of initiatives.
So, you know, as Dr. Radovich mentioned before, our program is really blessed to have amazing genetic counselors in it. So we have Leanne Stout and Cindy Hunter right now who take very good pedigree of our patients. We, of course, worry about patients who harbor inherited predisposition to cancer, as this is very impactful. But you know, with the advent of any technology, interestingly, always comes new obstacles, but also opportunities. And as you can imagine, as we're doing very deep and comprehensive sequencing of our patient's tumor, what we begin to identify is that a pretty high portion of patients also were found in the context of doing this to have inherited mutations. So we have found that somewhere over 10% of patients actually carry a variant in a cancer predisposition gene. And this is regardless of pedigree, meaning that while we typically use standard guidelines of age, tumor type, number of family members who have tumor, we found a decent proportion of patients had a cancer predisposition gene without really the evidence support testing for it. And so this has been, you know, again, a very interesting obstacle and also opportunity. Now, as we think about the patients that we do sequencing with, again, our goal here is to find a drug target. Importantly, though, as alluded to earlier, having sometimes an inherited germline mutation is also now a good drug target for many of our drugs like PARP inhibitors and so forth. Now, because many of the patients that we currently do sequencing on have metastatic disease, understanding that they have a predisposition to cancer may not matter all that much for the patient. This may change as we begin to sequence patients earlier in their journey. But what is critical is that these patients who harbor a predisposition mutation also have family members, many of which are healthy, who may now also carry that gene and be at risk for future cancers. So this has really brought about an interesting opportunity to think about how do we begin to find those family members and do the appropriate cascade testing for them.
So in summary, I think it's pretty clear that next generation sequencing and genomics is really a powerful tool to interrogate the cancer genome. As we talked about earlier, publicly available resources are invaluable to the study of cancer genomics. What we know is that cancer genomics continues to be advanced in its ability to develop better methods of detection, prognostication, drug development. But I think hopefully we were able to convince you that sequencing, informatics, clinical implementation has really ushered in the advent of true precision medicine for cancer clinical care, where we tailor therapies for each individual patient.
Yeah, and I just want to, take a second to congratulate all of you. I mean, I think you've picked an incredibly important field. I think what we're seeing is that the intersection between somatic genomics and germline genomics are really at an unprecedented place right now. And so the critical role that you guys will play in that in cancer medicine is a phenomenal one. So really excited for your career.
So we just thank you guys for the opportunity to be able to lecture you today. Lecture to you today. If you have any questions, please feel free to reach out to Dr. Schneider and I. But we also know your Professor Leanne is there as well, who's our amazing genetic counselor for our program, can also speak to our program as well. So we thank you for your attention, and we wish you the best.